Together with Pepe, we decided to give you a brief insight into what we're doing in 2 areas of biomedical research. One thing is providing materials for heart muscle tissue regeneration, and the other focus today will be on peripheral nerve regeneration. And I will show you what we actually can do with our types of materials. But before I come into these details, I would like to actually introduce silk a bit in more detail and about our recombinant silk technology. So if you have a look at silk materials, and we are specialized in spider silk, but you can also expand that to silk from silkworms or other insects. Many silks are suitable for biomedical applications, and the reason for that is that silk is a protein-based material, so it's biodegradable. As you can see up here, you can actually use protein-based materials to load and release biological factors, which is a benefit for wound regeneration or any kind of tissue regeneration. We can implement binding sequences like the very well-known integrin binding sequence RGD in order to actually get a cell interaction with the silk surfaces. Silk are very well known for their proper mechanical properties. They actually do not show immunoreactivity inside the body, and this is very surprising. But a huge benefit of using silk materials over other materials, since the human body obviously does not recognize silk as some foreign material and it does not react towards it. And in that context is also the lack of toxicity, which means we can actually really safely use silk materials inside the human body. I'll come back to that in more detail a bit later. So if you look at the natural sources of silk, as I mentioned, we are focusing on spiders, but there is also a lot of other arthropods, mainly insects, that are also able to form silks, like the very well-known silkworm, Bombyx mori, but also bees or wasps, lacewings, they produce silk. And they produce silk with different properties. And this is very important to know, that silk is not silk, but there's a huge variety. All of them are made of proteins, and if we have a close look, we see the different silk structures that can be adopted. The common theme between spider silk and silkworm silk is the presence of beta sheet crystals, and they have a huge impact on the mechanical properties, as you can imagine. In the bee and wasp silks, we see a lot of helical silks, so completely different protein structures, and of course then these silks have different properties. And the same is true for the cross-beta silk that is made by lacewings. So you see here 3 different types of structures, and obviously they're used for different purposes. And you see the purposes underneath. The world experts in silk production very likely is a spider, because spiders can use multiple silks, up to 7 different types, for producing webs, making cocoons, nests, and so on. So they are the specialists, so they can actually decide which type of silk they use for whatever application. In silkworms, the silk is mainly used for making the cocoon, so protecting the offspring during the transformation from the worm into the butterfly. Wasps and bees, they use their silk for housing, so they actually make composite materials together with wax to make their nice housings where they live in. And lacewings use silk, for instance, for breeding, so they produce egg stalks, and the egg actually is protected at the tip of these egg stalks from predators. So you see different types of applications: prey catching, offspring protection, housing, breeding, and this comes along with different properties that you need for these applications. Cross-beta sheets like the lacewing egg stalks, they are bending stiff. Yeah, this comes from this explicit structure. These helical structure, this allows really the incorporation into composite materials with waxes, because here you can easily provide hydrophobic surfaces that are necessary to interact with the wax. And we will focus now here on the batter sheet structures as we find in spiders and silkworms, and the main focus will be on spider silk. This is what we're working with, and just to give you a very brief introduction into spider silk, you see here a badminton racket, a speed badminton racket, and we have here spider webs just turned in, and you can play with this racket. And this is very surprising because the individual fibers, they're really thin, so they're between 3 to 5 microns. And this gives you, at a first glance, already an idea that it's mechanically strong. And this is obviously a natural property because the web has to be strong to actually hold up the impact of the inflying breath. Then, if you have a close look at the surface, you don't find microbes. So it's obviously somehow bacterial, repellent, and I come to that later in more detail because we just had recently a publication on that topic. It's well known since centuries that spider webs can support wound healing. They do not cause inflammation and they do not cause allergic responses, at least not to our knowledge. So combination of really excellent properties, especially if you think about biomedical applications. Now, I told you already that spiders are experts in making silk because they have huge variety of silks, but there's also a huge variety of spiders. So far, more than 48,000 spider species exist, and we just recently identified a very new species. One PhD student of mine found this species in Colombia, and she had the privilege to name it, and she named it after her German language teacher in school. So we are now very happy that we also identified a new spider species, but this is not our main topic. Our main topic is the silk. And as you can see, different spiders, they produce different type of webs. Yeah, they not all look like this web. Sometimes they are actually located in such a boat, wooden frame. Sometimes they're free hanging, sometimes they're a bit like messy, 3D, 2D, whatever. So you see different types of possibilities how to deal with the material, and obviously all the materials will differ slightly. And especially the Madrid group is very well known for mechanical analysis of the different silks, and they found out that indeed there are differences in the silks by different spiders, and this of course makes to me a lot of sense because they're used for different kind of applications in different kind of economic niches. So are spiders dangerous? I'm always asked that. And here is a sign that I found in Australia in the Brisbane Museum in Queensland Museum in Brisbane. This is the cause of death in Australia in 2013. And you know that in Australia, there's the most toxic species, and there's also the most toxic spiders there. But are they dangerous for people? Well, if you look at the numbers in 2013, roughly 44,000 people died of cancer, 30,000 on heart disease, 2,000 just fallen off a tree or a ladder whatsoever, 700 in car accidents, and so on. 4 were bitten by snakes, 2 by sharks, and 0 by spiders. So spiders are not dangerous. Spiders run away. So whenever you approach a spider, it will not attack you. So don't believe what you see in B movies, yeah, so that the big tarantulas go after you. No, they're not. So we are not their prey, and they try to avoid us. I'll promise you that. So if you look at the classical orb weaver, so these are the spiders with the most different types of silk, like the European garden spider, and this is one of our working horses in the lab. You see that indeed they can make up 7 different silks. So they start with the frame, which is the so-called major ampulate silk. And then actually this is accompanied by the capture spiral. This type is called flagelliform silk. This is just the mechanical setup. In order to keep the prey in the web, it needs some glue, and this type of spider uses a liquid glue. There's also other spiders, they have a dry adhesive wool-like structure, but these spiders here, they use a liquid glue, which is called aggregate silk. And this actually manages that the prey stays in the web as soon as it's getting in. Then there's the supporting silk, the minor ampulate silk. there is something to attach it to the surroundings. And then you see additional silks here for egg case shielding, for prey wrapping, and so on. 7 different types of silk, all have different properties, as you can imagine: sticky, aggregate silk, not sticky, the other silks. I will focus on the major ampoule silk because this is the best known silk. We not only work on major ampoule silk, but everything you hear from now on will be about the major ampoule silk. So the material has extraordinary properties. Why don't you see applications for it? Well, that's not true anymore, because I will show you that indeed there are applications there, and there are commercial products available for everybody. I will come to that in a minute. So first of all, I would like to introduce you our technology, because there is some problem with spiders. They are cannibalistic. And we call the technology Spider-Man technology, and I know Spider-Man in person, and you will see that in the movie. Oops, I have to go back. If I can start it, yeah. Yeah, so this was my son. What is the problem with spiders? Well, the problem with spiders is they're not a problem for us humans. They're not killing us, but they killed themselves. So most spider species are cannibalistic. So it's a big problem to actually keep a lot of them at a small place. In order to get a lot of material, you need millions and millions of spiders and a lot of co-workers, and this is a very expensive feature. So therefore, for any commercial product, using the natural material, using natural spider silk is not a good idea. In order to circumvent that, you can think about biotechnology. And biotechnology started in the 1980s to produce proteins in different hosts. We call that recombinant technologies. So you transfer the gene from one organism to another organism, and this other organism produces then the proteins. This is also a bit tricky because all genes are encoded with the codon usage, and this is different in spiders in comparison to typically used hosts in biotechnology, in bacteria. And therefore, we had to actually get around that. And what we did, well, we took the spider silk genes, we put it in a plasmid, and then we changed it. We made engineering, molecular engineering, to adopt the codon usage, to make it a bit better for the bacteria. Then we actually introduced this plasmid into bacterium, and then we can do large-scale fermentation. And this runs now in huge facilities, and actually nowadays companies can produce up to hundreds of tons of spider silk proteins just by using bacteria. The point is the bacteria only produce the molecules, right? They do not produce fibers. So this is another big question. But as mentioned before, this is not the main topic, but I wanted to show that indeed you can nowadays buy products for everyone. So this armrest for these watches is the very first full 100% protein fiber scaffold. So this is actually made of our spider silk proteins. You can buy that through the online shop of Omega. And there's a lot of cosmetic products, because even without having fibers, you can use silk to get smoother skin, better hair, whatsoever. And you see a lot of products that you can buy even here in the drugstore, in the pharmacy, and in Germany, it's available everywhere. And I think some of the products are also available in Spain. We sell them to South America, South Africa, Australia, Asia, and you can buy these products everywhere. So nail polish, So this is a tick removal coating. This is for coloring your hair, skincare, whatsoever. A lot of different shampoos, a lot of different things, which actually has our technology inside. But this is not the topic. Now I would like to focus on medical applications, and this is what we at the university are really interested in. And as I mentioned, the bacteria, they produce proteins. So what we have to do, well, we first have to process these proteins, and this is where now the engineer side comes in. So far we were molecular biologists, right? Now, actually, we get into the details in processing. And the very good news is that you can process spider silk proteins pretty much into whatever you like. So this behaves like classical polymers. So you can make fibers, of course, as in nature, but you can also make capsules and forms and nonwoven meshes, hydrogels, films, particles, and so on. We could show that more than 10 years ago that it's possible to make all these different types of morphologies. But the big question, of course, is A, do biotechnologically produced silk proteins have the same features as the natural spider silk? And B, what happens if we process them? Do we change then maybe some properties? And in other words, are all these morphologies also biocompatible? So what you have to do is to get into more detail, you have to severely test the body response. And I can assure you that no matter what we tested, what type of morphology in whatever normed test, we showed no immunogenicity, so no antibody response. No toxicity, no skin irritation, eye irritation, mutagenesis, and so on and so on. So it's a very safe product, and it was CE certified in 2014. So this actually allows us, since more than 6 years, to actually use it inside the human body. And the first product will come on market probably within the next 2-3 months. So we have all the approvals now at hand. So let's see what happens if we make a silk surface. Best to investigate is a flat film and see how human cells like it. Well, first we started not with human cells, we started with mouse cells, mouse fibroblasts, bulb 3T3 fibroblasts. So these are skin cells from mice. And to our surprise, so this is a tissue culture plate, and this is the silk fiber, the silk film. We saw that the cells don't like to stick to the surface. So if you make a proliferation test, so if you look over days, what happens with the cells, you see that they actually, agglomerate, they still are in a spherical shape, they're not attaching the surface, they're not spreading out, and they're not proliferating on the silk surface. But they do, of course, on the tissue culture plate. This was a very first indication that in this state, so in the native state, the spider silk is probably pretty much like a cell repelling surface. But that's not bad, because sometimes you want to have that inside the human body, that the body cells cannot recognize the surface. So we thought this could be a clever idea to protect implants. And this was the very first setup many years ago. And you see here, the publication was in 2014. And now the product comes on market because these are silk-coated silicone breast implants. How to coat a silicone breast implant? It's quite easy. You just take this implant, you dip it in a silk solution, an aqueous silk solution, I have to say. And silk molecules are amphiphilic. So what happens is they immediately assemble on the surface and they make about 1 micrometer thick film. So we get then a shielding, a coating of this implant. And to make a long story short, well, first we tested that in vitro, then we tested it in vivo in rats. Now we actually went through the complete clinical trials with humans. And there is a lot of patients already in Europe running around with our implants. And therefore we get now the approval to get it freely on market, and this will start soon to get it on market. Here you see the benefit. So this is just a plain silicon implant, and this is the silk-coated implant. And you see a clear difference, even here in rats, that silicon surfaces, they're very attractive for human cells, and for rat cells in this case. So what happens is the cells go to the surface, and they would like to destroy this foreign material, but silicon is pretty strong, so it's not biodegradable. And what the cells then do, they start to encapsulate them. So actually, they start to make cartilage surrounding this implant, and they start to make a capsule. For the rats, that's not a big issue. For human beings, that can be an issue, especially if you're a breast cancer patient, because you have a depressed immune response system. And therefore, these capsules can be really causing severe side effects. Because what might happen is an inflammation, and in about 25% of the breast cancer patients that got a silicone breast implant after their surgery, this implant has to be removed because it causes problems, and this is of course not nice for the patients. In contrast, if you look at the silk-coated implant, you see there's no coating. There's actually just tissue growing around. You even see vascularization, and this makes this coating very successful. Here you see what happens with the cells, and here we just had some DAPI staining, which stains the cell nucleus, and some collagen-1 staining after 7 days of cultivation. Uncoated silicon, you see that here the fibroblasts, they grow, they have a lot of collagen production. The silk-coated silicon, as before, they're not even spreading out, they're not proliferating, they're not making collagen. And this is the explanation why we don't get capsule formation and we don't get fibrosis. So we tested a lot of factors in first in rats, then in humans, and you see that most of the inflammation factors go down. So this is always in comparison with the non-coated silicone implant. You see that a lot of factors, they're dramatically reduced. There's only one factor, the CD4, that is slightly increased. But if you look at the factors, all the others are negative, which means they're all much lower than in the uncoated control. Inflammation factors, fibrosis factors, TGF-beta1, which is a very important marker for capsular fibrosis, severely reduced. Contracture markers, fibroblast to myofibroblast transdifferentiation. So this is an indication of wound healing, pretty much reduced. So we have a long list, and this is a very positive, because this led us to the model, which actually was now confirmed in human beings, that if you have a silk coating, and this is here just done for make it much clearer, because obviously we will coat the entire implant for the real stuff. But here we just coated half of the implant to show you the differences, what happens on the surface. On the silk surface, we have no cell proliferation, reduced extracellular matrix synthesis, no fibrosis, reduced contracture. So this looks much nicer than the other part. And this nicer surface, of course, has the benefit that we have less severe side effects. We don't see any fibrosis, for instance, in the patients. Now let's go back to the main topic. In the title, I promised you to talk about heart muscle tissue repair. So big question is, can we repair a broken heart, so to say? Yeah. What happens if the heart muscle is damaged? And this is a very common thing. Just in Germany, we have annually around 7 million heart attacks. Not all of them might lead to death, but what all heart attacks do, they block some of the blood vessels in the heart muscle. So the heart muscle is not innervated anymore, it doesn't get oxygen. And this actually leads to the death of cardiomyocytes, so the heart muscle cells. And cardiomyocytes have one drawback. They cannot migrate. So whenever you have a damaged zone, here shown in yellow, heart muscle cells cannot re-enter the zone and they cannot regenerate the muscle. And therefore, we always have scar formation. And you might now be able to imagine that if you have a scar in the middle of a muscle, you will have a severe impact on the function of this muscle. Yeah. So even if you survive the heart attack, there is some side effect because the muscle is weakened by this scar that is right in the middle. Yeah. So the idea is to support the heart muscle. So far, it's done by patches. So something is put on the top of the muscle to support the contracture. Our idea now is, can we actually really replace this scar tissue? So remove it by surgery and replace it with a real regenerated heart muscle tissue. That's the idea. So for that, we have to overcome some problems. One problem is to find the right material. We need a scaffold material where the heart muscle cells really like to be on. And one such material is fibronectin. And with fibronectin, this works very nicely in 2D, as you can see here. So heart muscle cells, the cardiomyocytes, they like fibronectin surfaces. They spread, they connect, they communicate. One problem with fibronectin is you only can do that in 2D. There is so far no technology available, and fibronectin is probably not the proper material for that, to make 3D scaffolds. But we need 3D if we want to regenerate a muscle, right? So therefore, we actually investigated silk, because as mentioned before, we can do with silks whatever. We can even 3D print that. Yeah. So the main question here was, does cardiomyocytes interact with silk surfaces? And to make a long story short, again, with our regular silk, they don't. Yeah. Remember, no cell would like to attach to the silk. So therefore, we had to actually further engineer our silks. And what we did, we introduced now positively charged amino acids. So on purpose, we actually changed the surface charge of the silk, which is typically negative, into a positively charged surface. The rest was the same. And out of a sudden, so we call this protein E80F4 for engineered Araneus diadematus, so that's the spider, fibrin 4, so this is the natural counterpart, kappa 16. Kappa stands for K for lysine, and we have 16 lysines in there. And if we make this engineered spider silk protein, you see that the cardiomyocytes now out of a sudden, they attach to the surface, they spread, they proliferate, they connect, and in the movie you see they even start to contract after 2 or 3 days. And we even could keep now beating cell cultures, cardiomyocyte cultures, for more than 60 days in culture. So as soon as they start beating, they're very happy, they like it. However, this is 2D, yeah? So, but this was the very first important step, to find the right material. The question is, can we also use other silk engineering? And as mentioned at the very beginning, we can introduce also cell binding motifs like the RGD. So we tested again now, so this is our working horse, the negatively charged protein, E80F4, you know what that means. C, this is glutamic acid, so they have a lot of negatively charged groups. And now we added the sequence RGD. And you see now all of a sudden, well, this surface is also good for cardiomyocyte adhesion. And even if we increase the concentration of the fetal bovine serum, you see much nicer proliferation. And again, make a long story short, cardiomyocytes start to communicate, and they actually start with a beating rate, and the beating rate is even higher than on fibronectin. So the white bars, they are with spider silk, the black bars are with fibronectin, and here you see the beating rate in minutes. And why is that important? Well, it's not just important that they beat, they have to beat in the right rhythm. And so we see here an increase in the beating rate already in 2D, and this is a very good sign, because this actually tells us the spider silk variant is even better than fibronectin. So if we now look for this first step, we have to go now further ahead because we have to go into 3D. And if we want to have a really 3D working muscle, what do we need? Well, we need vascularization. So we need blood vessels to grow into the silk. And for that, we hooked up with a team from Erlangen, and they are specialists in a so-called AV loop model in rabbits. AV stands for arterial venal loop. So actually they cut open an arteria and a vein, and then they artificially connect it, and they put it here in such a Teflon pan. So you see here, actually the arteria is coming in, and then it's going over into a vein, and it's in a Teflon pan. And inside the Teflon pan, now you can place a lot of materials. And if they are actually good materials, the vascularization from this AV loop into the material will start. And here we tested our spider silk. It's typically in the rabbits for 2 weeks and for 4 weeks. And then it's checked if after 2 weeks or 4 weeks, any new blood vessels are grown. So this is how the explant looks after 4 weeks. And this is what happened. And to our surprise, we see a lot of vessels growing out of the AV loop. And in this histological picture, you see a lot of newly formed blood vessels. All the black dots, these are new blood vessels that have been formed. So they're artificially colored to make them visible. And all the black dots, this is new material. These are new vessels that have been formed inside the silk material. So next step is done. Actually, in vivo, our material will be vascularized. And this is very important because if we have no blood supply to the cardiomyocytes, they will die. So we need that to take place. Another big issue is infection. And this is a study that has just recently been published in Materials Today. Of course, in all these tissue engineering approaches, we always have the problem with infections. We have a lot of pathogens. We have yeast, we have different bacteria, we have multi-resistant Staphylococcus aureus, and so on. In our respiratory system, in our intestine, everywhere. Yeah? So we're flooded with microbes. And of course, they cause problems if we would like to make tissue regeneration. So what we need is to prevent them from growing into any new kind of tissue. And typically this is done with antibiotics. But as mentioned, some Staphylococcus aureus, they are already resistant to most commonly used antibiotics. And also you cannot give antibiotics forever, right? So what you actually will need is a microbe repellent scaffold material. That would be the best. And you can do that by structuring surfaces. Yeah? Nanostructuring of surfaces has already been shown to be microbe repellent in comparison to microstructured surfaces, because they are actually well suited for bacterial adhesion. So nanostructuring is the key, but nanostructuring will also repel, again, all the human cells. So we are again in the problem. If we generate a nanostructured surface, we would like to have now bioselectivity. We would like to have the body cells growing on top, but at the same time having the bacterial repellency. And this was something that we investigated, though the whole study took 8 years, no kidding, 8 years to get the results, what we can do with spider silk. And here you see, again, our working horse, our negatively charged silk, flat films, and structured films, because we wanted to find out whenever we have surface topography, if this might actually accelerate or support bacterial growth. And here you see one of our control materials, which is polycaprolactone, a very often used polymer in biomedical applications. Here we just tested bacteria like Staphylococcus mutans, and down there we actually tested a yeast, Candida albicans. And as you can see, nothing is growing on the silk surface, no matter if it's a flat surface or if it's a structured surface. In contrast, on the polycaprolactone, you see actually huge areas covered with bacteria or with yeast, and this is what we call a biofilm. So we see immediately after a few days biofilm formation. So we already could show that on flat surfaces, spider silk works very well, and this, of course, accompanies what I've shown you before with the fibroblast studies and with the silk coating of the breast implants. Nothing likes to stick to it. So now the table is set. So we have a material that is bacterial repellent. We have some engineering in the silk that allows us to attract human cells, specifically the cardiomyocytes. We know this works in 2D. The next step is to go 3D. And for that, we have to go now to silk hydrogels. And this has been shown almost 10 years ago that we can make hydrogels out of spider silk. And this is just some SEM pictures to have a glance inside. And we see a porous structure. And the pores are fairly big enough to actually harbor human cells. So we thought these hydrogels are very well suited. The next step was, can we actually give these hydrogels also specific shapes? And for that, we tested robotic dispensing. So what you do is you put this hydrogel in any kind of capillary, and then even pneumatic or with a piston or with a screw, you actually press it through a needle. And if you have a good hydrogel, it starts to flow when actually the pressure is set on, and it immediately hardens if it comes out. So this is called the shear thinning behavior. Yeah. And then you actually can make precision 3D scaffolds. If you have a good bio ink, this is what we call a bio ink that has this behavior, that it's hard without any shear force. You press it, it starts to flow. Then it's good bio ink. And you know this behavior from toothpaste. I mean, toothpaste is not perfect because it also shows some instabilities later on, but in principle, if you have toothpaste in the tube and you open the screw, nothing happens. You have to squeeze it that it flows, right? And that's pretty much the same principle here. Okay, so can we do that with spider silk? This was the first test, and you see here a 3D printing in real time. So you see it immediately hardens as soon as it leaves the needle, and you immediately can start with the next layer. Mm-hmm. And the first idea was, well, now we 3D print the stuff, and here we 3D printed something that looks like an ear, 64 layers, and then we actually inoculated cells on top. Mm-hmm. The drawback of that system is, and this is for all kind of tissue engineering approaches, that the cells have to go into the scaffold and find the places. So especially with the cardiomyocytes, I've shown you before, they're not migrating. So if we just would put the cardiomyocytes on top of such a scaffold, they will never enter it. They just will sit on top. So therefore, we had to actually overthink that kind of story. And the idea was very simple. Well, if we just place the cells together with the materials in the scaffold, that would be the best option. And this is what is called biofabrication. So the simultaneous printing of the hydrogel with the cells. So we mix the cells with our spider silk ink, and then we actually print that together. And out of a sudden, the cells are in place. They don't have to migrate anymore. And the first thing we looked at was, do the cells survive that? Because if you just print cells without any scaffold, they're dead. Most of the cells die. And here you see a live dead testing after 40 days. And you see a lot of green dots, and probably you see non-red dots. A red dot would be a dead cell. All the green dots, they are living cells. So we have a survival rate here in the printing process of more than 99%. So actually the silk gel is not only the scaffold here where the cells sit in, but it also shields the cells during the printing process. So it shields the cells from the shear stresses, from the elongational stress that appears down here at the needle. And this actually allows us now to 3D print cells with the scaffold, and they're surviving that. Now let's go back to our bacterial story. So I showed you that the surface of the silk is microbe repellent. How about now in the hydrogels? Because if you have here a bacterial manifestation, that could be also very severe. And you can see very nicely that, again, our spider silks, and here you see our working horse, the working horse with RGD, they actually see no bacteria or yeast manifestation. In contrast to gelatin, which is very often used in 3D printing and biofabrication, Bombyx mori fibrin, so silkworm fibrin, that does not have these repellent features. And even if we modify this silk here, if we remove the charges completely, omega-16 is uncharged silk, then you see that we lose some of the ability of repellency. But be aware of, here's a break in the y-axis. So this is numbers of magnitudes higher in silkworm silk and in gelatin. And this is how it looks like. So actually what we did, we actually floated these hydrogels with bacteria and yeast. We incubated that for days. And then we just washed it. And you see that both spider silk hydrogels, they're absolutely clear. There's no single bacterium in there. And the silkworm hydrogel and the gelatin hydrogel, they're actually grown over with the bacteria and with the yeast. So what does that mean? Well, this means now we could do co-culture experiments. So we could actually put fibroblasts and bacteria into the scaffold at the same time. Then incubate that, and then actually check what happens to the fibroblasts and what happens to the bacteria. And the experiment is shown here. So the fibroblasts, they survive very nicely. So we have around about 95% survival rate over 2 weeks. So the fibroblasts are fine. And again, we have here an axis break, yeah? And look at these numbers. There's undetectable amounts of bacteria in gray and in yeast. So even in the hydrogel, this system works. What does that mean? Well, with spider silk, especially with the RGD variant, we now have a bioselective material that allows us to selectively grow cells on top and very selectively repel microbes. And we right now look at a lot of pathogenic relevant strands, multi-resistant strands, and so far we have not found any bacterium or yeast that can grow on the silk surface. Well, the story continues, but these are very exciting findings for us. And the reason is silk has a nanostructured surface. Remember, this is one physical principle that has been shown also by others that works in microbe repellency. So we can repel microbes just through the nanostructure on the silk surface. And if you want to find out more, this is published in 2 papers, Materials Today and Data in Brief, and they both combine the study that we took over 8 years, as I said. Last story, I make it short. Can we also repair peripheral nerves? Now, different topic, yeah, different story, but this is also something that we're very much interested in. So if you have peripheral nerve injury, so I'm not talking about the central nervous system, I talk about peripheral nerves. You cut off a finger, yeah, so you cut off a peripheral nerve. So nerves can actually, in principle, regenerate naturally. But this only works if there's a very small gap, yeah? Because you have then loose ends of nerve fibers and they have to find each other again. The further you get apart, the harder it is that they find each other. So therefore, if you have a bigger gap, there's just one probability that you actually get an autologous nerve transplantation. What does it mean? Well, they take some nerve, typically from the forearm, and took that out and replace actually the damaged side with this autologous nerve. The problem is you have a loss of sensibility at the donor side. That's one thing. And of course, you have not unlimited numbers of nerves that you can take out of one patient. So this gives us some limitations. Therefore, a group at the medical university in Hanover, they started to use spider silk, and they took spider silk from spiders. Yeah, so major ampullate silk fibers from spiders. These were supposed to be guiding structures for the nerve cells. Then they embedded this in matrigel. Matrigel is a gelatin hydrogel where they implemented Swan cells. And then they put this whole thing in a cava vein of a donor rat. So they removed all the cells from the vein, and then they put the spider silk fibers and this matrigel inside. So that was the setup. That's the vein. These are the spider silk fibers, and here in between you see the matrix of matrigel. And this looks good. So they actually tested that also in vivo in sheep, and they could span 60 centimeter defects in nerves with these, what is called a nerve guiding conduit. Yeah, so this is like a highway for nerve cells that they can connect again the loose stumps one to the other. And you see here the neuronal cells that they grow and they connect and so on. One drawback is you need a vein, and obviously you probably don't want to have a rat vein in your body. Matrigel has some problems because there are batch-to-batch variations in the performance. And as mentioned before, a natural spider silk is not that easy to harvest at large scale. So what we try to do is now using our technology, our biotech silk, to make nerve guiding conduits. And we also wanted to have a faster approach. This approach takes roughly 6 months. So if you want to span centimeters, the cells have to grow in, they have to connect, to proliferate. This takes a long time. We want to have a very rapid technology that allows us to actually get the nerve back to function quite quickly. And for that, we thought about self-rolling systems. So if you combine spider silk with chitosan, you have a 2-layer system that is responsive to liquids, because if you actually now add here cell culture medium, the chitosan will expand differently than the spider silk film, and you will get a self-rolling. So you actually can create out of a flat film a tube. And the benefit of that is that before you do that, you can actually implement other things. You can actually implement like spider silk non-woven mats. You can have also collagen cryogels. You can actually fill this stuff with whatever you like. We add the nerve cells, and then you actually have a nerve guiding conduit, which is ready to go within a few days. That's the setup. It also has recently been published. This is how the setup looks like. So you see, that's the filler. In this case, it's a collagen cryogel. And this is the self-rolling spider silk tube. And you see everything is nicely packed, and the fibers are aligned inside. And of course, we have the cells in place. Same principle as with the heart muscle regeneration. We put the nerve cells already along the structure. So we don't have to wait until they grow in and find their place. We put them where they should work. That's the idea. And then you see that the nerves nicely align along the channels in the cryogel or along the fibers, and they get functional. We also can use collagens for that. And this is a technology that we developed a few years ago, a microfluidic setup for spinning collagen fibers. And I wanted to show that to you because this is also a very unique technique to make endless collagen fibers with unique properties. All you need is collagen solved at a slightly acidic pH. You have 2 neutralizing solutions, and then you have the setup where actually the collagen solution is not getting in contact with the wall of this fluidic channel. And then you can make fibers like that endlessly. You can make knots in them, so they're mechanically quite stable, and they're much more stable than any kind of other collagen fibers. We have a higher Young's modulus, we have a higher strength, we have very good extensibility, and we have a low diameter. So these collagen fibers, they can also be implemented in such a nerve guiding conduit. You can even scale that up, and the scale up is running, so we actually can produce now kilograms of that stuff. We can even braid it, we can make new kind of structures for any kind of application. But back to the neural regeneration, these collagen fibers that we produce with the microfluidics, they're also very well suited for neuronal cell growth. And again, the neurites and the axons, they grow along the fiber axis. And here you see them much better. So we can actually pre-align very nicely the neurons with these collagen fibers. Second setup that we used, I've shown you before the self-rolling films. We can also use silk nonwovens per se and roll that up and then actually bring in the collagen fibers. So this setup is more similar to the setup of the Hannover group, but instead of having a vein, a cover vein, we have now here a silk nonwoven, and we have collagen fibers instead of the silk fibers. And this setup also works quite well. We see that the neurons, they actually really make neuronal networks. They interconnect, and you see here the connection points between the neuronal cells along the fibers. And we can even measure in vitro now the signal propagation through these conduits immediately 2 days after cultivation. 2 days versus 6 months. And I think this is a huge improvement. So this is our technology that we would like to bring in place. And of course, this is only done in vitro so far. So the next step will be to do that in vivo and to see if we really can bridge damaged nerves, peripheral nerves. And with that, my time is up. Come to an end by thanking all the people. So here you see my group. Well, this was before Corona. I do not want to change the picture because right now it would be, you won't see the people because they're so far off. I would like to thank all the sponsors of the studies, our companies that support us a lot, our partners, I mentioned some of them along the way, international partners, Bayreuth partners, national partners, our alumni. And finally, I would like to thank you for your attention. And of course, I'm happy to take questions if there are any.
Well, thank you very much, Professor Scheibel, for your justifying my initial remark about an extremely formative and informative presentation. Absolutely. And with regard to the questions and due to the limitations of this format, I would suggest that, well, there might be two options. Either using the chat, and there is at least one question now in the chat, but if somebody wants to ask you directly and to have the opportunity to talk to you, I would also be willing to open the microphone of this person. So there is, as I say, one question right now, and while there are more coming, it's from Manuel Antonio Oria. And the question is, if the silk by itself has a low cellular attachment property and low cellular proliferation, as said previously, how is it possible to create an hydrogel in which the aim is to harbor cells? Is the silk modified in order to alter this property?
Yeah, so very good question. And I hope, so I also see in the chat that my microphone had some problems. We sometimes have, I apologize. So I hope you can hear me now nice and clearly. Yeah, so this is the RGD variant. So we need to have the RGD modified silk. Because with the non-modified silk, well, the cells will be inside, but they will not actually spread out. They will not proliferate. Yeah, so it has to be modified with RGD.
Well, let's see if there are more questions.
I have a question. Can you hear me? I'm Jose Jalife.
Yes, please proceed.
Yes, Professor Scheibel, this is a very interesting presentation. It's a huge amount of work, and I really congratulate you for such an accomplishment. I'm interested in the heart, and my interest is in trying to understand how cardiac cells interact with each other. And tissue regeneration has been an important area of research over the last 10 years in the heart for myocardial infarct, and really bringing new materials into the field is a very, very important area of research, which is of huge interest in the field. However, and what you presented to us sounds very, very interesting, but the devil is in the details, as you know. And one of the things that has happened in the field is that even though some investigators have been able to introduce cells into a heart to fix the broken heart, the problem has always been, and there's beautiful data in the literature in monkeys and various other species, particularly in monkeys, that the cells do integrate, but the problem is that they generate arrhythmias. And so they generate arrhythmias and the animals die of arrhythmias. And that has been a problem that people have not been able to solve. So do you have any comments about how you would go to prevent this problem and to bring the field forward in a way that would be safe for the patient? And also, I would be interested to know which kinds of cells you would like to integrate into the materials. You want to do induced pluripotent stem cell derived cardiomyocytes, embryonic stem cells, or any other type of heart cells. Thank you.
Yeah. Yeah, so thanks for that. I mean, absolutely confirm what you said. I mean, the really big problems are way ahead of us. So far what we did, so the published story is mouse cardiomyocytes that we directly prepared from the heart. And I fully agree that to get really in the future, and I'm talking about in 15 years or so, that is my guess, a functional heart muscle that really works in vivo, we have to be aware of that it's not only cardiomyocytes, right? There's also other cells. There has to be communication. There has to be a very clear communication also between the other cells that are involved. And what we try to do is actually to get selective materials so that we actually really on demand can distinguish which type of cell is located where in the tissue. That is sort of the plan, but we are at the very beginning of that. And of course, the route towards that is not working with mature cardiomyocytes, but we started now with iPS. And so human iPS cells, which was also a bit tricky. So for 2 years, we did not get that running, but now it's running well. So we can integrate the human iPS cells, and we actually can differentiate them into cardiomyocytes in 3D spider cell scaffolds. This works. These constructs, they beat. However, we need a lot of cells. So right now we work with about 5 million cells per milliliter. So that is one of the big issues. We need enough cells to get started with. Of course, beating rates are far off what we will need then in the final construct. So this is also something we have to work on, how to stimulate the cardiomyocytes, how to get them mature. So a lot of unanswered questions. I'm just a materials developer, and we work close together with our heart muscle specialist at the University of Erlangen Clinical Center, which is Felix Enger. So he is the heart muscle specialist, so coming from the medical side, and we try to provide the materials, and we try to adopt the materials, engineer new materials to get step by step into the right direction. But I'm absolutely with you that's the big issue that probably will not be solved within the next, I would guess, 10 to 15 years.
I agree with you, and I thank you for your presentation. I think it is very stimulating, particularly for the young people who are hearing us. I think this is an interesting way to go, and I thank you.
Thank you very much.
Well, we have another question by Conchi Serrano from the Material Science Institute in Madrid. And she says, in which range is the Young modulus of silk foams? And how do you maintain the silk roll and the chitosan silk rolls together without opening?
Yeah, so thanks for that question. So the silk forms that we use currently, they are thought for skin regeneration. So this is what we do there. And therefore we need Janus moduli in the range of kilopascal. So we're talking about between 80 and 100 kilopascal Janus modulus in the forms. And they are made by salt leaching. But of course, we can produce forms in other ways, and they then have different properties. But the forms that we currently use are for skin regeneration in the kilopascal regime. Yeah, and with the silk, chitosan silk roll, well, as long as it's wet, it will stay in this conformation. And since we intend to implant that into the body, it will not get dry. Yeah. If you dry it out completely, then it will get flat again. But this is not an issue because as soon as we have packed everything, we would like to implant that, and then it will not get dry anymore. So therefore, it stays in its shape. And then, of course, the endothelial cells come in, and they also make endothelial cell layers. So the other idea is that the silk supporting material, and I forgot to mention that, it will be biodegraded over between 18 to 24 months. In this rolling tubes. So it will be replaced then by actually natural tissue. That is the idea. So we just need to have this in place for a few months, and then it will be gone anyhow, and then the supportive structure comes again from the regrown natural tissue.
Sorry. Now there is another question by Miguel Vicente, so I'm going to open his microphone. I think it's open now. Now, Miguel Vicente.
Yeah. Can you hear me?
Yes.
So Thomas, thank you so much for such a fun and whimsical talk. I mean, this was actually beautiful. So we are interested in cell migration mostly. And as you know, neuroblasts actually migrate on glial cells that extends like these thin, long processes that look very much like your collagen fibers. I mean, have you checked if neuroblasts actually migrate on your fibers or they just get attached there? Thank you.
No, yeah. So thanks for the question. Well, here I've shown you the already attached structures, but we also have the setup where we have the pre-rolled tube, and there we actually have the neuroplasts coming from outside. So they actually grow along the fibers. So they can also grow along the structure. So both is possible.
Okay, great.
Okay, thank you. Now we have another question by Daniel Gonzalez from CTV, and he asks, did you check if bioprinted cells establish cell-to-cell contact? This should be important to establish intercellular communication through connexins and transmission of cardiac electric potential.
Yeah, so very good question. So I've shown you the connexin connections in the 2D structures. We also right now running the same experiments with the 3D structures, but they just have to evaluate the data so far. So in 2D that works, yes. For 3D we have tested it, but it's not evaluated yet. Yeah, of course, this is very important. So the next step for having these 3D structures is that we also will apply electric patches outside. So we would like to stimulate the heart muscle cells now with electric potentials from outside. And for that, we right now generate a bioreactor. So that actually allows us to grow this piece of silk with the cardiomyocytes, and it gets then electrically stimulated during the maturation system. And right now we set up this bioreactor through traditional 3D printing and try to implement everything in a place. This is what we're currently working on. Yeah, so it's a very good idea. Yeah.
Now there is another question by Manuel Antonio Oria, and provided that silk structure is highly stable and it can be used for biomedical purposes, for how long is the silk material going to last in the organism? And in addition, if this material are degraded, is it possible that this material serves as a base for the deposition of organism amyloidogenic proteins in order to form amyloid?
Okay, very, very good questions. I start with the last question. No, it's not the surface for other amyloids, and I know what I'm talking about. Before I went into the silk business, I was working for more than 10 years in the amyloid business and in the prion business, so mad cow disease and so on. And of course, we tested a lot of amyloids that we have here still in our drawers. So silk is very sequence specifically inducing fibril formation. And as soon as you add something else, it does not work. So from our view and from our side, it's very clear that silk is not causing, at least in vitro, and we have never seen any complication in vivo so far. The other point, and therefore I started with that, the other point is that silk inside the human body is degraded, well, depending on where you put it, yeah, and in which morphology, more or less quickly, yeah. So we will not have any silk depots that stay there for years. It might cause trouble later, but the silk is degraded. So now it depends where you put it. And one important thing to know is that spiders recycle their webs by eating that. So if you have digestive enzymes, silk is degraded within minutes. And we tested that. So we tested stomach fluids and intestinal fluids. In stomach fluids, acidic pH, the silk is stable for weeks. In intestinal fluids with pancreatin, it's digested within 5 minutes. Then, of course, for us, more important, because we don't want to eat this silk, I mean, you could do that, but, well, not necessarily. We are more interested in wound healing devices. So, of course, we investigated how are wound proteases interacting with silk. And there, it really depends on the morphology. Particles are very rapidly degraded. So if we use particles for drug delivery. So for them, it takes about 2 to 3 days to be fully degraded, yeah, because you have a very high surface to volume ratio, and this gives a lot of attachment sites for the proteases. If we have films, they are much more stable. They are stable over weeks and months, and with the Preston lens, because this was one of the questions, for the Preston lens, we see that after around about 9 months, the coating starts to get thinner. And after about 18 months, the coating is gone. But then it's fine, because the side effect is fibrosis, which is the medical bad thing, so to say, only takes place between month 3 and month 9. So if the coating is stable more than 12 months, and that's the case, we prevent the fibrosis, and then the implant is fine. And it's well integrated into the human body. And then later, the silk will be degraded. The most stable structure is the fibers. Yeah? So they probably last, and there's study by Fritz Vollrath, so one of the pioneers in silk, spider silk studies. So they could show that it's stable for 2 or 2.5 years, but then it's also getting degraded over time. However, it really depends where you place it in the body, under the skin or really to an organ, because we found out that only very selected proteases like the MMP2, so the metalloproteases, the wound healing protease 2, in combination with elastase, can digest the silk. If you have the individual enzymes, none of them has any effect in vitro. So you need explicit mixtures of different enzymes, which you don't find everywhere in all the wounds. And this, I think, makes this situation very special.
Okay, thank you. We have, I don't know if there is any more questions. If not, well, once again, I would like to thank Professor Scheibel for his extraordinary attack on his time. And let me finish with just two personal remarks. The first is that I hope that we will be able to meet in person in a non too distant future. And the second is that I hope that Bayern München does a good match tonight, but hopefully not too good. So thank you, Thomas, and hope to see you soon. And now,
thank you very much. I'm looking forward to our next meeting.
Okay, so now we have a 2 minutes break, and then Gustavo Plaza will be the chairman for the second session. Thanks to you all.